Okay, this is Dr. Jack Kruse, man who hardly needs any introduction at this point. He's a neurosurgeon, trained also as an oral maxillofacial surgeon, became famous for his writings and lectures on quantum biology, in particular, the effects of light on metabolism and Neolithic disease. Also a fixture in the Bitcoin community. Also, I don't know if he really likes this or not, but becoming much more well known amongst politicians. Knows President Bukele, was formerly advising Bobby Kennedy's campaign. He's now the HHS secretary. He's been controversial. He's been revolutionary. He's been disruptive. But as time goes on, most important of all, he's been right. So, Uncle Jack, it's an incredible honor to finally meet you.
Great. Glad to be here.
I found you back in 2017 when I was a resident at Mizzou, a medicine resident. I joined your community and have been following you ever since. And certainly this was long before you did the Rick Rubin podcast, long before you did Danny Jones. And of course, now you talk about more than just light, but that still is the main thing you talk about. And for someone who's kind of dumb like me, I can capture and understand the blue light side of things. So as a physician, as an internist, the first question I wanted to ask you is, in order for us to truly understand medicine and disease, especially chronic disease, are we going to have to become quantum physicists?
I don't think so. I think it's actually pretty easy to understand. The problem is when you go through, what I would call standard medical training, nobody's having you read the journals and also the papers that you really need to understand. Like one of the big problems, I think, in centralized biology, centralized science, centralized medicine, I'm grouping those all together, is that there's a huge biochemistry bias, and most of the people don't even realize that bias exists. And it's pretty easy to see once you point out a couple of key features to people. Like the one that I always like to go after is, some of the guys like Thomas Siegfried, who tells people who have cancer, hey, just eat a ketogenic diet. And, you can do amazing things with things like tumors like I treat, which is GBM, which is glioblastoma. And that's really disingenuous because diet can't fix a quantum problem, meaning what light causes. And the reason he says the things he says, the reason you were taught what you were taught in medical school, the same thing that I was taught in medical school, is because we didn't understand that Archimedes' lever was actually light on the biochemical boxcars that we learn about. And I casually point out to all of them, especially when it comes to things like, diet, that if you look at cytochrome 1, which I think most people know, no matter if you're a biophysicist or biochemist, it's NAD, NADH. It gets talked a lot about everywhere. But one of the things that never gets talked about is that it's got a 340 nanometer emission spectra, and then it's got an absorption spectra, and then it's got an emission spectra in the red and the blue, and everybody casually dresses over that. And I'm like, well, do you think evolution or God makes mistakes by doing stuff like that and doesn't put those things in? Or maybe is it that maybe they're there for a reason that we still have not appreciated yet in centralized medicine? And of course, my perspective is it's the latter. And we don't understand that everything in biochemistry ties back to light. Now, where the story gets really interesting is when you explain to people why metabolism links back to light. And, I got into a lot of scraps probably 10 years ago with guys like Nick Lane from University College London. But now Nick Lane is really on my side. Yeah, he's on my side of things because he's beginning to realize that all the metabolic pathways we learned about in biochemistry, they all form, spontaneously. You can actually put them in a test tube, put the right nutrients in, and guess what happens? They run in cycles. The interesting thing, there's no control over it. And Nick has begun to realize that the biophysics is actually the key to which pathway gets chosen. But where does this go back to? Well, Nick is also into evolutionary biology. His main thing is he's been trying to figure out the story behind the ATPase and the story behind Luca. And it turns out what people have forgotten is actually how life began. It's an evolutionary story. There was 2 domains for a long period of time on Earth, for 3.8 billion years ago. And then magically, something happened on Earth before we get to this big event called the Cambrian explosion that I think every doctor learns about, where all of a sudden, overnight, we get 32 phyla in the eukaryotic domain that shows up out of nowhere, in evolutionary history about 600 million years ago. So the question you have to ask yourself is, what happened between 3.8 billion years and the Cambrian explosion for this event to try to make sense? And it turns out it's something called the Great Oxidation event. And when you understand in that period of time, things move from 0% oxygen to 21%, which is kind of where we're at now. The story of us as probably the most distal animal on the planet is we have an organ that I specialize in, the brain, that's an obligate normoxic aerobe, meaning that it works best when we use the TCA cycle. Turns out, when you look back at life, the first 2 domains, they were obligate anaerobes. They were functioning in a completely different environment. So between those 2 things, this story about oxygen becomes really important. And when you point out to people that the TCA cycle basically is a cycle that's based around oxygen levels that are really high, you start to ask yourself the question, well, what happens when oxygen for some reason is not very high around us, and can it be turned off some way? And this is where the rubber meets the road. It turns out when you look at oxygen carrying capacity, which every doctor knows is tied to hemoglobin, hemoglobin can only carry oxygen in the +2 state. When it gets to be in the +3 state, which is methemoglobin, it becomes incapable of carrying oxygen. And most doctors know that that's an issue. Most doctors, however, have no idea how to truly treat or diagnose methemoglobinemia, and most of them default to learning, look at the pulse ox. And it turns out if you look at a regular pulse oxes can't tell you if someone has methemoglobin or not. And the reason why I'm bringing this up to you now, why it's really important, if your patients are in front of things that change the oxidation state of iron, they can be fully conscious in your operatory, in your clinic, and they're walking around with between 5, 10, 15% of methemoglobin in them. What does that mean? It means their brains and their organs can't use the TCA cycle. And this is the biggest conundrum. And what happens, even though there's 21% oxygen around all of us, your mitochondria become incapable of using oxygen when the oxidation state is changed. And it turns out light is capable of changing the oxidation state of iron in every heme protein in your body. And when you learn that, that's when the game begins to change, because then you start to go, wait a minute, you're telling me that this can happen without us eating food, carbohydrates, fats, lipids. The answer is that is absolutely true. It's axiomatically true. But guess what? Nobody in medical school learns that that's true, because they never get one biophysics class anywhere in medical school. And to prove this point, to show you just how bad this is, about 4 years ago, when you and I heard all the stories about people going into the hospital, getting intubating and getting killed, one of the things that I realized immediately, it ties back to the story I just told you. I said, we've got a bunch of hospitalists and ICU doctors that have no earthly idea that most of these people with either a viral infection or some other organ system problem are walking around, they're hypoxic because they have methemoglobinemia. And I went back into the hospitals to prove doctors that I was right. And I showed them their pulse ox and how it didn't work. Then I took some blood out and did peripheral blood smears. And I said, take a look at their peripheral blood smear. Doesn't it look like, These are primitive cells. I said, well, it turns out that's what happens when you have hypoxia. Things go backward. They're like, well, I didn't learn that in medical school either. I said, well, it's funny because Robert O. Becker, twice nominated for the Nobel Prize, published in the 1960s that when bone regenerates, we use red blood cells that de-differentiate under the power of hypoxia and a blastema. And they all looked at me like I was crazy.
Yeah.
And eventually what I did, I showed them that all their patients in the ICU fundamentally were hypoxic, even though the SAT rate didn't show it. And I said, if you start to treat these people like they have methemoglobinemia, you'll never intubate them. The reason you guys are intubating these patients is because their SAT rates are dropping into 92, 88, 84. And even though they're able to talk to you and they're conscious, you are being trained by an algorithm in medicine from critical care. So I had them put on a different pulse ox. And I said, take a look at this. I said, you guys all learned in third year medical school that people who have COPD, if you give them oxygen, you decrease their drive to breathe. I said, what do you know about methemoglobinemia? And all of them looked at me. And I said, just so you know, every single person here has it. And you may not know this, but when you get a viral infection, it actually causes damage to the intermitochondrial membrane. And when that happens, they get hypoxic, and that makes methemoglobin. I said, so you don't need to intubate him, because when you intubate him, you just turned a case from hypoxia into ARDS. And when that happens, then you're going to French fry their liver, you're going to French fry their kidneys, you're going to French fry all different organ systems in their body, and you're not going to know what's really going on because they can't talk to you because you intubated them when they didn't need to be intubated. I said, so you have to learn not to pay attention to the SAT monitor, because the SAT monitor is not accurate in someone who's got methemoglobinemia from, a pathologic problem. It can be from trauma, it can be from a virus, it can be from bacteria, it can be from really anything. But the number one cause in the world today is screens. Screens actually do that because screens change the oxidation state from +2 to +3. And when you go to +3, you can't carry oxygen. It's that simple. And where did that lesson come from? It came from the great oxygenation event that happened, 2.4 billion years ago. And I explained to all of them, I said, if you understood that this story is actually how we got eukaryotes, it's actually how archaea and bacteria joined. I said, you'll find out that oxygen being pumped into the environment here behind me, 2.4 billion years ago, was an electrical resistance problem, meaning that oxygen was toxic to the first two domains of life. So what did the first two domains of life do? The first cause of actual cancer in the world was the endosymbiosis event. That's actually why they formed together. It's because oxygen was pumped in tremendously. And if you're hypoxic, which these two domains of life were, what did it do? This electrical charge built up, and as the electrical charge built up, the impetus for that to happen was exactly what you see in a warbird shifted mammal. What do we do in a warbird shifted mammal? This is where Thomas Siegfried's right. It fosters more a cancer state. Why is that? Because it takes normal differentiated stem cells and pushes them back atavistically to a previous evolutionary format. And then when you add oxygen into the mix, the oxygen actually fuels the cancer. I said, what people have forgotten in the endosymbiosis event, there was no heme proteins that were present when archaea and bacteria were present. So the first two domains of life had no answer for oxygen toxicity until one thing happens. What's that one thing? It's the evolution of heme proteins. And what does a heme protein do? Well, you need to know that a hemoglobin, because that's the one most doctors understand, looks exactly the same as chlorophyll. Chlorophyll was innovated on Earth 50 million years before the Cambrian explosion. And the thing that most doctors don't get taught is why was magnesium selected and put in a porphyrin ring, just like iron is placed in a porphyrin ring. And it's pretty simple. Magnesium has a redox stable oxidation state. It's +2. It's got 12 electrons. So when you put it inside that cage, it means light can't affect its oxidation state. So what did that do? They took CO2, created more oxygen, and that's how you got the whole food pyramid. That's where every single food web ties back to that process. 50 million years later, right at the beginning of the Cambrian explosion, evolution kicks out the idea of heme proteins. Where does it get this idea? From the story I just told you. Oxygen now is going from 0% up to like 10 or 15%, and oxygen's toxic. So they take bacteria and archaea, put them together, and the two lines to stay away from dying come up with an evolutionary brilliant maneuver. We're going to take the porphyrin ring, remove magnesium from it, and put iron in it. Why did they choose iron as the first heme protein to be innovated? Because iron has two oxidation states, +2 and +3, as I told you already. +2 allows the animal or the organism to bind oxygen. That means so that it can control it, so it's not toxic. And the +3 state, actually, you can bury it in other heme proteins so that it doesn't cause a problem. So what do I believe is the first heme protein that's ever innovated by the joining of archaea and bacteria? It's cytochrome C oxidase. Why do I believe that to be the case? Cytochrome C oxidase actually does something. It's, if you look at my fingers, I don't know if you'll use a video or not. This is, I got to do it this way. This is NAD. This is the beginning of the mitochondrial membrane, and this is oxygen. In between here is where all the cytochrome proteins are. And on that, we know that there's 30 million volts of potential charge from this electrical resistance that oxygen creates. Why? Because oxygen is electronegative. It pulls electrons across this thing, just like you learn in medical school. But the problem is when this 30 million volt charge breaks, say from COVID, from an infection, from bacteria, from trauma, it doesn't matter what the cause is, it breaks. That means that electricity can get out. So why does nature come up with the first heme protein that's likely CCO? Because it makes deuterium depleted water by reversing the process of photosynthesis. Why did nature pick that? I told the doctors that I taught in the ICU during COVID, the reason why is, if you know anything about physics, water that has no electrolytes and no deuterium in it doesn't transmit any electricity. It's a perfect insulator. In fact, many people know it as a perfect Faraday cage, but we don't tend to talk about that kind of stuff in medicine too often. But when I showed them the physics of what deuterium depleted water was capable of, you begin to understand now why Nick Lane in his book Power, Sex, and Suicide said it's kind of weird that we have this 30 million volt charge, but it doesn't seem to affect the mitochondria. Well, it turns out, what's the second part of CCO that you learn, but you never understand the evolutionary purpose? This was the purpose of coming out of the GOE. It's called apoptosis. So that if this breaks, you want to get rid of that bad engine right away. Why? Because what did nature learn when you had aerobic glycolysis, which is known as the Warburg shift, and this breaks, you would have a joining of two cells. So that's the first cancer state. So CCO, apoptosis, blocks that from happening. It's the first maneuver in evolutionary biology that makes sure that the two joining of forces, which are now eukaryotes, don't develop cancer. And what people don't realize when I say this stuff, if you re-engineer modern mammals today who get cancer from looking at screens or looking at LED lights, what do you have to realize? We have already innovated CCO in us. But at the beginning of life, they didn't have these heme proteins. So that would have caused massive unfettered growth of the joining of both bacteria and archaea so that you would eventually get that. And what happened over the process, I guess you would call it of empiricism, testing it, we came up with the idea of heme proteins. Why? Because then we could control oxygen at low levels. And as we gained control, what were the control mechanisms that are obvious today in our patients, but we don't get taught this in biochemistry? Pretty simple. What do you know about oxygen? It's paramagnetic. You didn't learn that in medical school. What does that mean? It's drawn to things that have a spinning FO head, meaning a magnetic field. That's the reason why breathing, is electromagnetic, meaning it's quantized. Okay.
Is that also the reason why you can generate exponentially more energy using oxygen-based production, like oxidative phosphorylation, than
In other words,
more
you
primitive?
actually build up an electrical resistance to use the power in the electricity to generate things. So if you think about what I told you before, chlorophyll is hit by light, and it is also a version of electrical resistance. Because what does it do? Light hits it, it stops, and then that electricity is then formed and goes to the Rubisco enzyme to make all the different foods that we have. So anytime light is stopped dead in its tracks, that energy becomes physiologically useful. So it turns out, well, how did we make the 30 million volts between NAD and oxygen useful? Here's your answer right here.
So when you slow down light, you create matter?
Well, not only do you create matter, but you create the possibility of electrical resistance distal to it so that these things can innovate solutions. But what I'm telling you right now, we're focusing on the first bit of electrical resistance. And it turns out that's the CCO enzyme, cytochrome C oxidase. Why? Because where does that cytochrome, where is it found? And this is important for you to understand. It's found right before the ATPase. So we're creating a huge amount of electrical resistance at that level so that that energy then can be distributed to other places to cause other innovations in the cell. And doesn't that make sense when you think about the story that Darwin could never figure out? Why all of a sudden did we get 32 phyla? Seems like overnight. Well, because guess what? We figured out we needed to use photobioelectric resistance to capture the light, to use it to do other things. And the interesting part is if life didn't figure out this event really early on, that means that the two domains of life would have constantly be in a state of joining forces and forming useless cells. Why? Because it couldn't make any sense of the power that it was harnessing from the environment that was pumping oxygen in slowly over, we think, 1.2 billion years. So things got more powerful. What was one of the next things that comes up from this CCO enzyme? Well, we make a thing called hemoglobin, which I think all of you know. Looks just like chlorophyll, except it's got iron in the middle. And then there's something even more amazing. On hemoglobin, most people forget this. There's another paramagnetic binding site for what? A free radical called nitric oxide. And what does nitric oxide do for oxygenated hemoglobin? This you should have learned in medical school, but I have to tell you, speaking to a lot of doctors, they tell me, yeah, I never got that information when I took hematology, that NO binds to oxygenated hemoglobin and decreases the amount of oxygen on hemoglobin. I said, well, does that make sense that nature would use something like that when oxygen is going from 1 to say 10 or 15% or up to 20, that there's got to be a cap so they can protect itself? And what did that cap eventually do? That's where the other cytochrome proteins are made. And the other cytochrome proteins further were able to make other magnetochemistry, which is superoxide, hydrogen peroxide. These are all chemicals that become electric resistance molecules that slow light down to do different things distal to it. And when you actually see what nitric oxide does, it also acts at cytochrome C oxidase, which is the fourth cytochrome right before the ATPase. So now you have a cap on how hemoglobin can work. And it turns out when nitric oxide binds to hemoglobin at a significant amount when we're hypoxic, guess what magically happens? The oxidation state of hemoglobin in your body goes to +3. That means that you can't carry oxygen anymore. That means that you can't use the TCA cycle. So why would this be beneficial in the story in your clinic and my clinic? When do humans have a period of their life where pure hypoxia is incredibly important, where they're mimicking the first two domains of life? That's called when you were in utero. When two cells, mom and dad, come together and you're trying to join them, What do you know about that environment? What does everybody axiomatically learn is true? I would say whether you're a doctor or not. You're inside a uterus, inside an amniotic fluid bed, and there's no oxygen getting in there. You're completely hypoxic, just like life would have been at the bottom of a hydrothermal vent in the ocean.
Mm-hmm.
And what happens for those 9 months? You go from 2 cells to a 9-pound baby in 9 years, don't you? You have incredible growth. Now, that growth is generally controlled, assuming that you truly stay hypoxic that whole time. Well, what happens if there's a change in your oxidation state from, say, things that your mother does or things that your father does? Well, that can create a situation in the child where they get a transgenerational epigenetic cancer. Okay? That's the bad stuff that you and I see. Now, the story that I just told to someone else that I think is really important, when you parse out this science and you go read Becker's book, like I mentioned before, so that everybody knows axiomatically the central dogma of biology that we all learned in medical school is bullshit. Becker found that in the blastema, that means when a bone is broken in both amphibians, reptiles, and humans, this is true basically from the Cambrian explosion up to now, because he covered all the animals that were in those 32 phyla. He found something interesting, that an adult red blood cell actually will dedifferentiate first to methemoglobin, then it'll get nucleated. What do you know, Dr. Bronson, about fetal hemoglobin? Doesn't it have a nucleus, and doesn't it have mitochondria? Let me ask you a question. Do any of your patients that come to see you in the clinic have that situation in their blood smear? No. So guess what? Here's an interesting conundrum, isn't it? And why should this stop you dead in your tracks? You go open up Becker's book and you're going to find out the first experiment that he did to find out that bone was solid state P&N semiconductor with 2 copper ions on it, and that it was all physical. He found that those animals healed their bones completely without any scars. Did you know that salamanders and frogs have nucleated red blood cells too?
I did not know that.
Yeah, most people don't ask the question. So this famous guy in biology history that you probably have heard something about, maybe not, a guy named Ernst Haeckel, came up with a term one time when he looked at small little embryology textbook pictures. It seems like everywhere from the Cambrian explosion, that when you look at human development, we have a tadpole phase. We have the branchial cleft stage, which, look like gills on fish, on amphibians. And you start to go, is that a story here that we're kind of missing in medicine? Have we kind of danced over this whole thing and not realizing that this whole story about oxygen controlling not only morphogenesis but also metabolism may play a role in why we may be developing chronic diseases today? So I'm going to push all your people listening to this. Hopefully there's a lot of doctors out there. When they go read Becker's book again, he said something very interesting, that when you look at salamanders, you look at frogs, you look at reptiles, you look at humans, once they repair their bone and regenerate it completely, remember, bone does not repair. You get told that in medical school, but Becker proved definitively it completely regenerates. So when you have a broken arm and it heals, I'm assuming no plates or screws, just old-fashioned regular bone break. When an orthopedic surgeon goes back in and does something else, there's no scar on the bone at all. That's true in humans, it's true in amphibians, it's true in salamanders, everybody else. Now, fast forward to my residency, and this is when endoscopes first come out. And an OBGYN sees this pregnant lady, and the pregnant lady finds out that she's got a baby inside of her. This is the 3rd trimester. The baby's got hydrocephalus. So these endoscopes were new. My staff doctor said to me, hey, we're going to do something cool. This is something we've never done before in our program. We're going to operate on this lady, and we're going to fix her child's hydrocephalus. We're going to poke through her belly, then poke through her uterus, and then go right over here next to the germinal matrix, next to the coronal suture where it should be. We're going to make a small little hole and go through the brain. And we're going to go into the third ventricle. We're going to pop a hole in the bottom of it, let the CSF from inside the ventricular system go over the subarachnoid space, and the child will be born and not have to have a shunt placed. And I'm like, now that's cool. I'm all in. I signed up for that, no problem. I got to do that. That's the first third ventriculostomy that I ever did in my life. This is like 35 years ago. What's the coolest part of the story? I get to see, because I'm still on my same rotation, I get to see the child born. And of course, I go up there. And when I see the child in the incubator, I ask the nurse to open it up. And I noticed that where I did the surgery, there was no scar at all. And then the child wound up having a scan done for some reason. I think it was because they were worried that the kid might have had a germinal matrix bleed. And there was no evidence that anybody had been inside this child's head. And I thought to myself, could this be the exact same story of what happened with Becker's salamanders? Because when did I operate on the child? The kid was completely hypoxic. It's the only surgery that I've ever done up until that time that didn't have an endotracheal tube, because it didn't need one because the baby was surrounded by amniotic fluid.
Sure.
And it dawned on me, I said, this means that we have incredibly huge regenerative potential when we're still in. Then I did a hard stop after this case, and I said, wait a minute. The two things in human that regenerate completely that I did learn about in medical school are bone and the liver. And then it dawned on me, that's where the umbilical cord attaches from baby to mother. And I said, that's where fetal hemoglobin's made. And I started to think about this, and I said, you know what?
This is not an accident.
No, it wasn't an accident. In fact, this is the story that I've been trying to tell you earlier, where the GOE, the great oxygenation event, is recapitulated in our physiology. And what's the mistake that we are still making in medicine? Because I know you know this as a guy who just trained. Everybody who listens to this podcast is going to know that most of the deaths that happened from COVID happened in ICUs and people that were intubated. Why? Turns out all those people that were in ICUs were just like this little baby that was in utero. They should have been taken care of in a hypoxic fashion. Why? Because that would have generated. As soon as we put a tube down them to increase their FiO2, guess what we did? They had broken intermitochondrial membranes, and as soon as we gave them oxygen, what'd that do? It pulled all the stuff across the garbage, and we made ROS. All of that ROS destroyed the rest of their organs, and that's why they died. Now, if you understand what I've just said to you in the last 20 minutes, this is the saddest part of the story. This is all provable and published, but no one in medical school is learning this. Yeah. Nobody in medical school learns that bone truly does not heal. It regenerates. And it turns out in utero babies do exactly the same thing. So that means when the baby's slapped on the ass and we cut the umbilical cord, it takes its first breath, that we need to do things differently on humans that are obligate TCA normoxic animals than we would normally. And we're not thinking about this because we're not thinking about the evolution that created this oxygen and nitric oxide system on hemoglobin. Why? Here's the craziest part of the story. Nitric oxide and oxygen are both paramagnetic. That means they're both drawn to that FO head. And it turns out that nitric oxide can be knocked off of hemoglobin to make it to go from +3 to +2 by doing what? Near infrared light. So guess what everybody should have had done instead of an intubation tube? Everybody should have had near infrared light, because guess what happens? We would have taken their blood cells and turned them from +3 to +2 so they could carry oxygen. They didn't need any tube. Got it? Now, they still would have had problems because, remember, you still have to fix the original problem, which is them having a viral infection that does this. But what was the answer to fix that? The answer to fix that was realize that cytochrome C oxidase was broken. So deuterium depleted water and methylene blue and things like hypertonic saline solution would have been the optimal choice for most of those people then. But why wasn't that on anybody's radar? Why wasn't it on anybody's bingo card? Because nobody got taught the story I just told you over the last 20, 25 minutes. Nobody realizes that our heme proteins are built to protect from an oxygen holocaust. And that's what patients in the ICU, I don't care what their problem is, whether it's brain surgery, cardiovascular, autoimmune conditions, you name it, even cancer. All of these patients have a problem with their ABCs. People learn about it in BLS. We learn about it in ACLS and ATLS. But we're not putting together this very, and I would call this a foundational cornerstone issue that's present in all of our patients. That means many of the patients that come to see you in the clinic, they have a methemoglobinemia that is undisclosed to you. And there's no way for you to pick it up unless you have a special post-ox in your office, because you have the ambient awareness that you learn this in medical school, which you didn't. And you didn't learn it in residency either. And there's very few places where you learn this. I happen to be fortunate because I did a lot of critical care neurosurgery when I was a resident. And that is how I learned about these special post-oxes. Because it turned out many times we would have people come in who had drowned in cold water, and we would code these people for a period of time. And one of the things that we noticed is that people did really well after we did supportive care and warmed them up, and we gave them methylene blue. That's part of the reason why neurosurgeons are experts in using methylene blue when people have a problem with hypoxia. Who's the other specialty that learns about this? And I would say not everybody does, not in every program. But the other
CT surgery, probably.
Cardiovascular surgeons. Why?
Yeah.
Because when they have somebody on bypass and pump, those people are hypoxic. Well, what do you want to do when you want to make them normoxic, when you're shocking their heart and they don't come back? Well, the reason why it doesn't work is because there's not enough oxygen in the system. Remember I told you about the electrical resistance of oxygen? It's the same story that's present here. And if you give somebody a bolus of methylene blue, and if the cardiovascular surgeon's really smart, they'll actually use hypertonic saline. The story that I told in Danny Jones' podcast, when I famously corralled Dr. John Oshner, who's Alton Oshner's son, we were actually doing a basilar tip aneurysm where we split somebody's chest open, and they had problems coming off pump. And I can tell you, that was the first case that Dr. Oshner ever learned about how to use methylene blue and hypertonic saline. He goes, I didn't know that those two things in combination could actually shock the heart. Why? Because after we clipped the aneurysm and we took the person off pump, what did we have to do? We actually had to give them methylene blue and hypertonic saline to bring them back. So that way, when we shocked the heart, before the chest was closed, that the patient's heart started beating and everything was fine. And, at that time, we're using ridiculous hypothermic, anesthesia technique at that time. This is back in the early to mid 90s. We don't do the surgery that way anymore. I mean, this is the reason why this is a lost art. But the reason I bring this story to you as an internal medicine doctor, I want you to know it wasn't that long ago that doctors were basically doing this stuff, and our patients truly were like being in a physiology lab, not with a dog or a guinea pig, actually with a person, a human being. And it turned out all the basic physiology that was in Becker's book was extremely useful, at that time in medicine. Unfortunately, that has not translated into critical care medicine, not in every place. And the reason I know that is because the number one place to go to die from COVID wasn't because the disease was bad, because we intubated everybody and gave them oxygen, when it turned out every single one of those patients were hypoxic and they should have been treated like bacteria or archaea in the GOE. And if we would have done that, they wouldn't have died. So that's
so
why
interesting.
It's important.
But that actually ties back, everything about light and energy ties back also to the explosion of metabolic disease, right? The obesity rate 100 years ago was 5%. Now it's like 42. It's 20% amongst children.
You understand what I said in other podcasts? I'll explain it to you. Every time we have 10% increase of tech spending, the NHANES rates for obesity go up similarly. Why? I just told you the answer. When you put somebody in front of a screen, you change their hemoglobin from +3 to, I should say, +2 to +3. So you're making the hypoxia worse, and it happens in wherever the electrical resistance is worse in people. So for obesity, where is that? It's in the leptin receptor. It's in the HPA axis. That's why it happens. Now, say if the target of the electrical resistance problem is in the habenula nucleus, then we get crazy people. We get
anorexia and stuff like that.
You get not only anorexia, but you can get bipolar disorder, you can get depression, you can get schizophrenia. And people don't realize where this biophysical paramagnetic toggle switch occurs, destroys the heteroplasmy ratio in the distal tissue, and that's the disease that your patient gets. And the greatest part of this story, the reason why it's so very important for doctors to understand Doug Wallace's work from the Children's Hospital of Philadelphia, if you know how to reverse that process and lower the heteroplasmy rate, guess what? As fast as the disease showed up, like it does in an ICU with ARDS, you can reverse the process if you know what you're doing. So that's the reason why 4 years ago, I went into ICUs and started teaching doctors this issue. And they looked at me and they're like, how the hell did you learn this? I'm like, well, I paid attention during evolutionary biology class about the Great Oxidation event. And I started to explain to people how this fundamentally worked. And I said, the number one way for us to be better doctors, we need to start doing peripheral blood smears on people that are hypoxic, because we can actually see the evidence of the dedifferentiation of the red blood cells. It's almost as if the patient is screaming out to us, do not give me oxygen, because if you do, you're going to cause organ failure. And if they stay alive long enough, you're actually going to stimulate oncogenesis in them. And that is one of the big problems that we're seeing already in people who took the COVID vaccine. It's not just from the SV40 promoter. It's not just from all the pieces of DNA in there. These people all have aerobic glycolytic metabolisms that have been shifted from all the things that have been done to them. And then when they go back and they're in a normoxic state and their cells are in an atavistic state, guess what magically happens? They get cancer. And this is the reason why this story was incredibly important, because when Becker wrote his original book, he told us about the microwave event that happened at the Moscow embassy and how 3 straight ambassadors died of blood cancers. And I always thought to myself, he's telling this story for a reason. I don't think he knows exactly what the reason is. I said, but in his work, he found out that 1/1,000,000,000 of 1 ampere current dedifferentiates these cells, and it's helped by the change in the red blood cell and the blastema. I said, why is it that these guys got blood cancers? And it turned out, guess where the problem was? It was from the pulse microwaves coming in from the Russians trying to teach the Americans a story. And when you read that in his book and you hear the story I just gave you, you start to go to yourself, so could this be the reason why we've seen this huge spike in cancer that no one has been able to solve since the 50s? Because we reached
processed foods and microplastics and shit like that.
Well, what they do is they blame food for what light's causing. And the reason why, guys like Thomas Siegfried, guys like Peter Attia, guys like Sean Baker, they only learn what you and I learn. It's only about carbohydrates and insulin. And then if they happen to pick up a biophysics paper like Nora Volkow wrote in 2011, just by putting a cell phone up to the side of your head, what does it do? It raises your blood sugar and it raises your insulin level up. Guess what? Light does that. Because what happens? Light changes the oxidation state. What happens when the oxidation state falls? You get pseudohypoxia at the mitochondrial level. And then what happens? NAD drops. Immediately, I just explained to you 30 minutes ago what happened at the GOE. That's exactly the same thing that happened. And when you begin to see that oxygen and light are the key to controlling the metabolic pathway we use, that should make you do a hard stop and go, if that's true, what Jack just said, then we should have an option somewhere on our integument that codes for UV light and for oxygen. And it turns out we do. We found it, 2009. It's called neuropsin.
Neuropsin, yep.
And guess what? When you see the whole story laid out, you start to go, wow, this is all discoverable. And you want to know something? Guys in first, second year, I mean, there's nobody that's learning all these pieces. Why? Because everybody that learns things in medical school have the biochemistry bent. And that's when I like to point out to people, if you want to know why Becker got canceled, 3 biochemists that ran the NIH are the ones that canceled him. Why? Because he was getting too close to telling people the truth that his whole regenerative loop from mammals was tied to an electronic, a biophotonic electronic circuit that actually heals us. And the thing is, they canceled him, but they didn't cancel the 2 people that worked on the side that destroyed it. That was Nora Volkow and this guy named Alan Fry. They were both DARPA scientists that showed how you could destroy the basics of how a cell works, but they never allowed anybody to learn that bone doesn't heal, it fully regenerates. Bone works by P&N semiconductors. It has 2 dopant atoms on it called copper that are electrostatically bound, which if you understood that, then you'd understand why the 2 Russian astronauts called cosmonauts in 1971 died in space of fulminant osteoporosis. And it turned out this story of what happened in space was an early predictor of what was going to happen in our ICUs now in 2025. It's what's happening in your clinic now. It's the reason why everybody's vitamin D level is low. It's the reason why Wolff's Law doesn't seem to work anymore. Why? Because the more of that EMF we pumped into that atmosphere, everybody is suffering from an oxygen holocaust. All of our patients are hypoxic to some degree, and we are impotent to see it because none of us have pulse oxes that work to measure their methemoglobin level. Nobody does blood smears anymore. I mean, that's the backup. If you want to know the truth, the backup to this story, you could start doing blood smears on every single one of your patients, and you're going to be magically shocked when you see those red blood cells that look like microcytic, iron deficient red cells. Or remember the disease that you learned in medical school called anemia of chronic disease? Guess what the cause of anemia of chronic disease is? Blue light non-native EMF. It's associated with every single chronic disease known to man. Why? Because of what I told you before. +3 to +2. They can't carry oxygen anymore. That's the reason why this happens.
So then is it fair to look at something like metabolic syndrome, which a lot of centralized doctors consider a mother disease to all kinds of cancers and even just obesity? Is it fair to consider that obesity is not really a disease of energy excess, but actually it's a disease of profound energy loss and inefficiency?
Well, that's exactly what obesity is. I mean, I did a TED talk that was pretty famous and got banned because that was the take home from the talk. And it got banned because the people in Big Pharma and DARPA didn't like that I was beginning to tell people the truth about what Becker really found in his work. And I think they were afraid that you would get onto the scheme. This is, remember, what drugs did they have in the pipeline at that time? They had the Ozempic drugs ready to go, even all the way back then. They knew that they were going to be able to print money because they were going to create a lot of fat people from the use of technology. They already knew it. The data was already there. The problem is they didn't want anybody to know it. That's why they shelved the leptin trials in the early 1990s. They found out that this was a big problem, and they knew about it. And they knew that the key cause was tied to this story about the oxidation state. And when Nora Volkow wrote her paper at NYU in 2011 and showed that blood glucose went up and insulin went up just from the use of a cell phone with no food, that should have caused people like, Thomas Siegfried and the other guy, I'm blanking on his name. He's a pediatric endocrinologist from UCSF. He had a very famous talk about fructose being the cause of obesity in kids. I'll think of his name at some point. But anyway, he was a pretty famous guy probably 15 years ago. Not so famous anymore. Why? Because he wanted everybody to blame food for what light and oxygen were really causing. Why? Because Big Pharma did a spectacular job hiding this basic information that I'm telling you by making sure that the leptin trials never went to full course. And if they would have went to full course, guess what we would have found out? That all of metabolism is controlled by oxygen tensions and by the light in our environment. And that's ultimately why neuropsin, melanopsin, encephalopsin, all these opsin proteins are everywhere where light needs to be signaled. And if you would have understood that, that means every disease, like the disease you asked me about, because I want to be accurate about this, metabolic syndrome is a disease of electrical resistance in the body, and you are losing that 30 million volt charge in all these different organs, and that's the reason why the changes happen. And remember, you know that the BP changes there. Well, what do you know about nitric oxide, my friend? Decreases your blood pressure, doesn't it?
Yep.
So aren't you starting to see the pieces and parts? What did I tell you about the liver? What was the main purpose of the liver in a baby? Remember, the baby wasn't eating. It was making fetal red blood cells. Well, what would you expect to happen in metabolic syndrome when you're hypoxic? The liver gets huge again, doesn't it? And that's exactly what happens in these cells. And why does that happen? The fat builds up in the liver. You get fatty liver disease because you can't use what, my friend? The TCA cycle to burn the fats. It's a consequence of this oxygen problem. And you have all these doctors, even some of the doctors that follow me, like Max Gulhane, convinced that seed oils are the cause of non-alcoholic fatty liver disease. And what have I been telling them for 2, 3, 4, 10 years? None of them will listen. It's all biophysical. And when you don't know the basics, that's how big pharma gets you. All it takes is for you to miss the key piece of data, and then you'll continue doing the stupid shit that they want you to do, selling all kinds of drugs to people and doing all kinds of things that make no sense. And as soon as you begin to see this laid out in front of you, you go, I always thought Jack was crazy. I always thought, he was outspoken. And I've said it on multiple podcasts. I'm still waiting for you guys to come prove me wrong. That's all I'm asking for. Because guess what? I've done the heavy lifting. I know the physiology in and out better than most of you. But the one thing I do know, I know how the biophysics works, and none of you do. None of you know about that paramagnetic tunnel. I've been warning you, too, on the blog. I've been telling you, why did I go hack the periodic table after I did that 18 months? Every single atom in us that's germane and important is paramagnetic. Like, I wrote a blog 12, 15 years ago about the inner mitochondrial membrane. I said, do you know what's really important about the inner mitochondrial membrane? 2 things. It doesn't have DHA in it. That should stop you because DHA is the only lipid that's never been replaced in 600 million years since the Cambrian explosion. So that you should ask the next question. Well, what key atom is big by not having DHA there? Molybdenum. It's also paramagnetic. And then you start asking yourself, how does this all fit? How does this entire issue fit? And then you begin to put the story together for people, and they start to see all these biophysical levers, like I like to call them Archimedes levers. And when you see how they do different things at all the different cytochromes and how they're the levers that pick TCA cycle, aerobic glycolysis, anaerobic glycolysis, gluconeogenesis, you start to go, wow, this means all the metabolism basically is waiting for instructions from light and oxygen to make the choice which thing to use. And that's the reason why, if Siegfried was right, if Peter Attia was right, that the Warburg metabolism is bad, then you as a doctor have a duty to explain to your patients why is it that the fovea of the eye and the gonads of both animals use a Warburg metabolism normally. You know what I'm going to tell you the answer is? Those are 2 places where light's present, that there's no oxygen there. Go look at the fovea. The ophthalmic artery comes in, it's a giant circle in the middle of your retina. There's no oxygen there. What else isn't there? There's no melanin either. And why is that the case? Because it's always photooxidized all the time. So that means it has to be turned over constantly. Doesn't that sound like what goes on in a baby when you're hypoxic? What's the same thing in gonads? What's the key with men? You know that men have a difference in their germ cells than women. They don't make any until they go through puberty, and then they make germ cells until they're dead. They make sperm like crazy. Well, if you're going to make sperm 400 million after every load you shoot, why do you think that the gonads where sperm is made have to be hypoxic? There's your answer. Because it's doing the same thing that happens in a baby. Do the heart stop now with ladies? You know, ladies are a little bit different. The first 2 phases of meiosis, they have all their eggs to begin with. What does that mean? Means that the granulosa cells around it are critical in keeping all the stem cells hypoxic. They get the oxygen, but not them. Do you know what hormone controls both of those things in male and females? CYP enzymes, heme-based enzymes. Oh, look at that. The heme story shows up right there again. So you have the break on the great oxygen holocaust. Guys,
Amazing.
So when people like Danny Jones ask me, Jack, why is every male have low testosterone? Jack, why does every female seem like they have PCOS? You begin to realize what the answer is. When you've introduced trillions of frequencies of non-native EMF into that ionosphere behind you, you're creating a great oxygen holocaust. And since we are the penultimate mammal that's built for normoxia with the TCA cycle, it shouldn't surprise you why we have 50% infertility. Because we are facing the same thing the T-Rexes faced. The difference is they saw their problem flying down from them at the Yucatan Peninsula. We've allowed Silicon Valley to create ours. And we're addicted to it as we're getting taken out. And people who criticize me are telling me I don't know what's going on, when it turns out they're the stupidest motherfuckers that people are listening to.
It's the Dunning-Kruger. So it's the artificial light not only makes us fat and sick, but stupid as well, right? We have a reverse effect,
when we say stupid, I want to be accurate for the audience.
I'm unable to discern the truth.
Well, the reason you can't discern the truth is because this is a Ferrari engine that gets what? 20% of cardiac output. What does that mean? 20% of the hemoglobin in your body that's supposed to be oxygenated. Well, what happens when that 20% isn't? Oh, so your brain is working on 15% is what you're saying. Well, can you have full cognitive abilities? You want to know what long COVID is now? I just explained it to you. Do you want to know what the Dunning-Kruger effect really is? Do you want to know why food gurus and biochemists, why I have such an ultimate disdain for them? Hopefully you're beginning to see now why I sit outside, do all my shirts, or I should say all my talks with no shirt on. Because when I do it like this, I am fully engaged with nature, making sure that this Ferrari engine always is in the +2 oxidation state. I never want to be in the +3 state. And when people post comments to me on Twitter, on LinkedIn, hey, can you dumb down the blogs? They just basically told me that they're not my target audience.
Yeah. Well, I've learned an incredible amount from you, but it's also kind of depressing because to me, there's nobody, you know, I have a textbook on metabolic syndrome that's very famous. They publish it every other year where they finally have a chapter on circadian rhythm, just one single chapter. But still, it's the same horse shit, the processed foods, the microplastics.
You want to hear the funny thing about the clock genes?
What's that?
You want to hear the funny thing about clock genes? You know the number one chemical that controls clock genes, what it's called? HIF-1 alpha. What does HIF stand for?
Hypoxia-inducible factor.
So when you actually see this,
That's
and then
amazing.
you realize
I didn't even know that.
Right. And here's a corollary to that. Do you know that the two nuclear circadian regulators are Rev, ERB, alpha, and beta? Both of them are heme proteins. Do you think that nature is telegraphing to all of us that this great oxygenation event was probably one of the greatest things ever for us to learn about if you happen to be a mammal that takes care of TC obligate normoxic mammals with a Ferrari engine in their head? Like, are you beginning to understand that no, you're not like an alligator. You're not like a crocodile. You do not have, there's a reason why reptiles had to have small brains. Now you're beginning to understand why. Because they had so many other amazing things in them that they couldn't run a Ferrari engine in their head. And then when you begin to start to see this, you go, wait a minute, Jack is saying to keep our oxygenation in the +2 state. This makes sense why we lost our hair from monkeys. Why? Because you needed to always stay in the +2 state to make this thing work really good. Got it? Like, there's so many different things here that we don't need new research. We just need to read the stuff that's been done and then put the story together, because Mother Nature is telling us an amazing story. I'll give you another one. Why do modern Americans, modern humans, get tinnitus, Meniere's disease, acoustic neuromas, cataracts, and vitiligo and all skin diseases? Why? If you look at any of the processes, a melanin sheet is always between the outside and the inside. Do you know that in almost all the senses, you go from 6-layer cortex to 3-layer cortex? Do you know what that means? That means that there's less myelination and less mitochondrial density in 3-layer cortex. So you'll find a melanin sheet anywhere where that happens to protect the situation. So how many times have you heard me say, if you've been a member since 2017, that you never want to put a nuclear power plant plugged into a house wired from the 1920s, because what happens? It oxidizes and burns up. What did I just tell you about the story about us? That we used to have 3-layer cortex in most parts of our brain through the evolution from reptile to I should say from amphibian to reptile to us, okay? That's also the reason why Huberman didn't know that we had melanopsin, an amphibian blue light detector in our brain. Well, it turns out any place we still have 3-layer cortex, which happens to be in some pretty important places that are tied to sensation, you begin to realize that nature put the third semiconductor there to protect it. Not only do you have heme protection, but you have melanin there to do the same thing. This whole system is built around the biophysics of light and oxygen. And it's really about time some of these doctors out there put their biochemistry books away and start to learn about biophysics. Why? Because if you want to solve the diseases that Bobby Kennedy's not going to solve, this is how you do it.
So I wanted to ask you about that. You at one point in time, you were advising his campaign. Doesn't seem like he really listened to anything you said, or at least what's going on there. What is your opinion on just the whole, what he's pursuing public health wise?
Well, I think he's a compromised candidate. I think he's been hampered by the people that he's in with. I think he's hampered by his own personal history. They likely have some dirt on him, but to be honest with you, even if the dirt turned out to be horrible, I don't think people would care as long as he did the right thing in helping chronic disease. For the things that he ran on when he was a candidate against Trump, these things were wildly popular. It's the only reason really Trump got elected. He got elected because of the Bitcoin bros, and he got elected because of the Maha moms. Now Bobby is completely, broken rank. In fact, the person that should be the most pissed off about this whole thing, I think, is Nicole. Why? Because Bobby Kennedy used Nicole as an ATM to get, into Trump's circle. And now I think several of the guys that are big time Cantillon effect, big pharma guys, specifically like Kennedy in Louisiana and Cassidy, Collins, all those people, they're being paid off by big pharma. They don't want this cash cow of the vaccine story taken apart. And we saw that the first challenge, it was really what I would like to call a test case for Bobby, is what happened in Texas. Do I believe that was totally set up as a psyops to see, was Bobby going to play ball like he told Kennedy or not? And what does he come out and say? Yeah, I think vaccination for MMR is the best thing for measles. That should have given everybody a hard stop and go, that Nicole's daughter, Echo, got autism and this is straight from Nicole's own mouth, that as soon as Echo got the MMR vaccine, she wound up developing severe autism. So when you think about that, he used her money to get in, and then this is the first test case, this is the problem that I have. Like, I think Bobby will do some good things on maybe the food and the red dye number 7 stuff, the stuff that, I try to warn people in the Danny Jones podcast that Callie Means and his sister were basically handlers. They're the handlers for Big Pharma, the handlers for the bankers, to make sure that Bobby was going to play ball with the administration on different things. They'll let them fix certain things, but only up to a point. And, do I think that there's nefarious reasons for that down the pike? Yes, I do. Do I trust him at all anymore? No, I do not. And I don't believe that he deserves our trust. Just like I don't think Bondi deserves our trust, or Kash Patel. We were told that people who broke the law in the past, specifically politicians like Adam Schiff and Nancy Pelosi and Elizabeth Warren and, Dan Crenshaw, all these guys that are making $200,000 but have $250 million in the bank, that they would go to jail because we'd find out that all the money they stole from the treasury and sent to Ukraine came back to them in their coffers. So they're effectively robbing for us. They are the people that Thomas Jefferson warned us about 250 years ago. But what's happening here, Dr. Bronson? When everybody's neocortex is in the oxidation +3 state, people are more interested in what the Kansas City Chiefs and the Titans are going to do in the draft than or what's happening in Starbucks or what's going on some stupid TV show, than really what's going on for their country being looted and robbed right in front of their face.
Yeah. What's the end game for all this? I mean, I guess I didn't have any hopes that he was going to do anything about the mRNA platform. We've had multiple very reputable physicians have their lives, careers destroyed. Dr. Paul Merrick, he's someone mentioned in the past. He's had his career totally destroyed. And it's obvious now that there's very serious problems with the vaccine. Like that's a fact. That's not an opinion. It's a fact that the mRNA vaccines are dangerous. But yet,
I think it's actually a bigger fact that most of the vaccines are actually dangerous, and that doctors need to start asking better questions of centralized science and also Big Pharma. In other words, we need to start to study vaccines just the way we study drugs. And until we start doing that we're always going to have a huge problem. Just remember what I told you. Every virus, every vaccine does this at the inner mitochondrial membrane. As soon as you know that means that it's a direct assault on the heme proteins in our body. As soon as you learn that and you're a doctor, you start to go, wait a minute, I believe that these vaccines are not causing a problem, but you know what? Jack's right. I don't do any testing for heme proteins. I don't know anything about this. That's all by design. If you don't know how to test for it, you don't know that it's going on. And how can I tell you that I know that I'm right about this? This is the reason why every single COVID patient was given an endotracheal tube who got admission to an ICU. Why? Because they knew what the eventual effect would be, that these people would die, and that death rate would make sure that people in your practice and my practice would roll their sleeves up to take the shot in the future. So when you ask me, what is the end game? It's about control and compliance. It's about bringing people back to the way society used to be when monarchies ruled and it was total centralization and everybody followed one rule. And as a doctor, you're a younger doctor than I am. I was in the transition state of where we went from doctors who could think to doctors who just practice by algorithmic medicine. Today, all medical schools create doctors who follow algorithms. They're not taught to think. Like, just think about the stuff that I told you here.
Yeah, that's definitely true.
The stuff that I told you here, you send it to any one of your colleagues, I guarantee you 99.99% if they're telling you the truth, will say, I never learned that light could change the oxidation state of iron from +2 to +3, and certainly never learned about the implications that this creates methemoglobin in them.
Yeah. I never learned much of this. I used to get yelled at by the unit clerk in the ICU back when I was a senior resident, printing off your old blog posts and some of your early Patreon posts, wasting hundreds of pages of printer paper. But that's where I'd sit. And man, that was it was very enlightening, but also very dark, because having some of this knowledge, particularly about the, easier stuff, light, it made going to work every day depressing, knowing that a lot of what we do is just harmful, at best counterproductive.
Yeah,
but in many cases, harmful.
It's harmful for our patients, but it's really beneficial for the profiteers, which is the big pharma guys, which is the hospitals, which is the big corporations. Remember, their billing coding goes up as ICD-9 and ICD-10 codes go up. So the sicker someone gets, the better it offers for them. And I try to point this out to people, and people hate when I say this, but the number one most profitable part of every hospital is their cancer service. So if you think that anybody's going to want to hear this lecture and where the cause of cancer really is, which is a light and oxygen holocaust phenomenon, no. MD Anderson doesn't want to hear this. All the nurses that got brain tumors up in Boston, I actually put a tweet out this week. I said, most of the doctors, the experts they're going to hire, none of them will walk in there with what they need to. They need to walk in with a spectroscope, and they need to walk in with a syringe to draw the girl's blood, and we can do peripheral blood smears on all of them. I can explain to you pretty damn quickly. Not only that, put them on a pulse ox and look for how much methemoglobin they have. You're going to find out that it's off the chain. But you know what? Do you think any of the experts that those big hospital systems are going to hire are going to do any of that? They're like, why would I get a spectroscope? Well, I just told you the reason why. If the spectroscope will tell you if they're +3 or +2, can they bind oxygen or not? If they can't, guess what? That's the first step to understanding where all their brain tumors came from.
Dr. Kruse, it's been an honor and a pleasure to have you. I wish I could talk to you for hours. I wish I could come meet you. I know you're on Instagram. Everybody is following you on X as well. Thankfully, the censorship on X, have you faced any censorship recently from Elon Musk's?
Yeah, well, Elon Musk is definitely shadow banning me. I'm definitely controlled opposition, but I haven't really come out strong against him yet. Why? Because probably the strongest thing I've said is I think he's the new version of Lee Harvey Oswald, that he'll be a useful idiot. Right now, he's a very useful idiot for DARPA, but there's going to be some day down the road where he's not, and then he'll be taken out just like, anybody else who's a useful idiot, with the government. And they need him right now, because right now they're using cell phones and screens in front of us to change our oxidation state, to make sure that we're obedient idiots. Eventually, it's going to happen from above.
Very depressing. Dr. Jack, I want to thank you so much. Putting blue blocking glasses on my kids was the best things I ever did. Help with their sleep. Obviously, we greatly limit the screens and Wi-Fi and stuff like that. So from a practical standpoint, but thanks so much for your time, and I hope we get to interact again sometime.
No problem.
In the future. I really appreciate it.
Anytime. Take care. It was nice talking with you.
You as well. Talk soon.
Right.
Bye.
Bye-bye.